Hi everyone, welcome to the second portion of the first exercise. And we left off in the prior presentation by discussing where the FASTA file is located, as well as the pre-computed results on our GitHub repository for this training course. So what we're looking at now is that GitHub repository in the single gene sequences directory. You see here's our FASTA file. If you're not familiar with FASTA files, this is what they look like. There's a header row where we have the name of the sequence, then the sequence itself, and it's just repeated as many times as there's a query sequence in the file. It's very simple, and this is the input file type that SeqScreen requires. So if you have a different type of file, you will need to convert it to FASTA before running it through SeqScreen. Okay, and then in the pre-computed results subdirectory, there's a number of different things, including the SeqScreen command that you can run if you are going to execute SeqScreen yourself. So you can just copy and paste this, with the exception of this particular flag. If you don't have internet access, go ahead and delete that. That won't impact your results for this particular portion of the course. So no worries there. You can use all of the other portions of the command when you execute SeqScreen with this input file. If you are not going to do that, these are the output files in the report generation directory that you can download and unzip. Take a look at everything that's there if you're interested in what would have happened if you had run the software. And this is more information about the sequences that were used for testing. But this is the file that I really want you to download and be ready to look at with me. So it's called Final TSV Report Single Gene Seqs, and it's a spreadsheet. Please go ahead and download that if you haven't already. To download something, you just can click on it and then download, or you can clone the whole repository, whatever's easiest for you. Go ahead and just grab that, get it onto your local system, and we will take a look at it in just a moment. Before we do, I'd like to orient you to where we're at in the SeqScreen workflow process. So this is the SeqScreen GitLab, and you'll find this workflow figure both on the README and in the wiki documentation, but it gives you an overview of within Nextflow what the different workflows are that SeqScreen is executing to analyze your query data. The first workflow is an initialization step. And essentially all that's doing is just making sure that your FASTA file is in a valid format. So is it formatted in such a way that there is a header row as appropriate with the sequence underneath each header, and there's no duplication in the naming of the headers, that kind of thing. It's checking to make sure that everything looks good so that it can be successfully processed in the downstream steps. What we're going to really focus on in this exercise is the SeqMapper workflow, which is kicked off in sensitive mode directly after the initialization step. If you're wondering what the different colors are in this figure, those correspond to different modes. So sensitive mode is green, fast mode is yellow, ONT mode is purple, and blue means it corresponds to all the different modes. So they all have those pieces. You'll notice a lot of green boxes in this workflow figure, and that's because not only does sensitive mode execute BLAST to give you more sensitive results, but it also has all of these optional pieces and extra modules that are going to give you more information about your query. All of that takes a little bit more time, a little bit more computational resources, but it can be worth it if you want to get as much information as possible from a query sequence. So when I have a small number of sequences that I want to analyze in as detailed a way as possible, I will always use sensitive mode. If I have a large number of sequences that I'm just trying to get through, I'm going to use fast mode. And if I have sequences that are long with multiple open reading frames within each query, I'm going to use ONT mode. So for this example, we have single gene sequences. We don't have a lot of them. I've chosen sensitive mode, and we're going to focus in on this part right here of sensitive mode in the SeqMapper workflow, where we take the query sequence and we align it in both nucleotide space and amino acid space. So bowtie 2 aligns in nucleotide space, and wrap search to aligns amino acids to this custom database of something we call BSET hits or BSET organisms. Essentially, it means any biological select agents or toxins that were listed in the US lists or the Australian lists, as well as commerce control lists. Basically, any kind of sequence that's publicly available that has been noted by a regulatory authority of being something that we should be paying attention to when we do this type of screening. So taxonomic lists, gene lists that are noted. We put them in this custom database and do some alignments. This is a sensitive alignment. It is not a specific alignment. So that means we're really just looking for anything that has similarity to a biological select agent or toxin. We are not saying that just because it does hit to this database that it is something that should be regulated or we should be concerned about, because there are genes like housekeeping genes and other types of genes that are shared by organisms that are closely related to those biological select agents. So it doesn't mean that we need to be concerned about it just because we get a hit here, but it is something worth noting. And so we're going to take a look at that next. Let's go ahead and open up that output file that I encouraged you to download. And then we will briefly give you an orientation to the different columns here. We have copied and pasted the output from a TSV report in SeqScreen into this spreadsheet. And that TSV report ended in pathgo.tsv. And it is exactly the same as the normal SeqScreen final report. It just has this extra column appended to the end of it called pathgo. And I like this because pathgo is an abbreviation for pathogenesis gene ontology. It is an ontology that was developed by Johns Hopkins University Applied Physics Laboratory under the IARPA and GCAP program. And Gene Godbold and others from our biocuration team collaborated with the pathgo developers to help review and refine this ontology. Essentially, within the SeqScreen framework, what pathgo means is if there is a pathgo annotation that appears with one of your query sequences, that means that there has been a manual annotation of that sequence for pathogenesis terms. So terms that describe why this sequence contributes to pathogenicity at the functional level. A human has gone through and reviewed that and recorded a PubMed ID that shares the experimental evidence for that particular function for that sequence. And there are not a lot of pathgo annotations just because of the amount of expert curation needed to assign these terms. Most sequences will not have a pathgo annotation. And in that case, you're just going to see this little dash there. If it has been reviewed by an expert but determined not to have any pathgo term assignments, you'll see a 0. So that, is also worth noting. When we look at these two zeros, I would expect these to not be genes involved in pathogenesis. And sure enough, this is a gene that's involved in the structural basis of the vaccinia virus. So it's not directly contributing to anything regarding pathogenesis. It's just how the framework of that virus is created. And then this has to do with tRNA, so basic processes, or you might call this a housekeeping gene or something that's basically not involved in pathogenesis. So that makes sense. And this is something that I like to look at. So that's why I copied and pasted this TSV report into this spreadsheet for us to look at together. Now let's go back and look at a couple other fields. The first is query sequence. So if you remember back to when we were looking at the FASTA file, that header information is now listed here. So these are our query sequences. I've named these in an intuitive way for the purposes of this exercise so you can see where the sequence came from. These are all publicly available sequences. Some of them have gone through and been codon optimized for different organisms just to show how the results may change or not change based on codon optimization. And if that is true, if it has been codon optimized, you'll see that here it's been optimized for gene expression in E. coli K12. Here, the same gene has been optimized for expression in Baker's yeast and so forth. So there is the query sequence name. Multi-tax ID confidence is another thing that I think would be good to pay attention to. And the reason for that is that when we look at the BSAT hit column, which is the output of SeqMapper, so SeqMapper's here. Remember that we're paying attention to whether or not something hit to the custom database based on mapping in nucleotide or amino acid space. And it's a binary yes/no classification. So yes, it did match to something. No, it did not match to something. Here we see a lot of yeses because we're looking at Bacillus anthracis genes, and those are genes that, or those are organisms at the taxonomic level that are on the biological select agent or toxin lists. So we see yeses. What's interesting is that there are genes shared among the Bacillus genus, and likewise, other threat organisms have genes that are shared with non-threat organisms that are closely related to them. And we see examples of this here. So I have highlighted in red a couple things just to illustrate this. These don't natively show up in red when you get your SeqScreen outputs or you copy paste things into Excel. Just FYI, I did this. These are genes not from Bacillus anthracis, but we see NCBI tax IDs here that correspond to a Bacillus anthracis strain. So this is Bacillus anthracis strain H9401. That's what this NCBI tax ID refers to. And then we see a colon and 1.0, meaning that this is a perfect match to this particular organism. It has high confidence that it belongs to this organism. But when we look at all of the other information in this field, we see that it has high confidence in other things too, right? It's equally shared across a number of different tax IDs. So that tells me that this is a conserved gene, likely among the Bacillus genus, maybe further. I haven't gone through and looked at all these different tax IDs, but I can tell you that a lot of these are Bacillus. So this is something that is shared between Bacillus anthracis, which is on the BSAT list, and these other Bacillus organisms. And so when we look at the SeqMapper outputs, we see yes, but that doesn't mean that this is a threat. That just means that this sequence has similarity to something that is being regulated at the taxonomic level. So that's a very important point. Hopefully that was clear and everyone understands that we're not working from the BSAT hit column to determine whether or not something should be concerning. We're just paying attention to that as a piece of information that we may want to consider when we're starting to synthesize our results and decide on how to interpret the final decision and results here. Okay, so we see a lot of yeses. I'm looking for some no's, so we can just talk about those too. All right, so at the very bottom, we have some no's. These were not matching to anything in the biological select agent or toxin list. And when we look at these, what you might notice that these all have in common is that these are eukaryotic organisms. So these are snakes, spiders, scorpions, and they produce toxins that obviously have some pathogenic effects because they're toxins. However, eukaryotic toxins in these cases are not on any of those regulatory lists. So they may show up at the functional level as having some toxic capabilities, but they are not going to show up in our BSAT hit column here. And that's really the distinction that I wanted to show you. Coming back to just the global idea here, SeqScreen is trying to give you additional information about sequences of concern. But while it's doing that and sharing all this functional information with you, it's also providing you with tax ID matches, with BSAT hit potential, and lots of other things. So those are things to keep in mind as we move forward and you begin to interpret outputs. So that's it. That's the basic premise of this first exercise, is just to become familiar with the SeqMapper outputs and how those show up in the final report and what they mean. Hope that was helpful, and we will move on to the next module. Thank you so much.